with us today. And so her talk will focus on personalizing dietary caffeine and health research, which I was saying I'm personally interested in because I think that many of us consume quite a lot of caffeine in a normal day and would love to hear more about this topic. So Dr. Cornelius is an associate professor in the Department of Preventive Medicine at Northwestern University Feinberg School of Medicine. And her research aims to couple modern high-throughput omic technologies to traditional clinical and epidemiological methods so that we can enhance our biological understanding of how diet and nutrition contributes to chronic disease. And so her specialty is, of course, the genetics of coffee consumption, caffeine metabolism, taste preferences, and other dietary behaviors. And we just learned that she actually did her postdoc here in Boston at the Harvard School of Public Health. So she might have encountered many of you at that time, or now through a few collaborations that are ongoing with Tufts. So welcome officially, and thank you so much for joining us. Take it away.
All right, thanks for the introduction. And again, thank you for inviting me to this speaker series. This is just two main components of my talk. I'll be first briefly talking about the traditional approach to caffeine and health research. And then the bulk of today's talk will really be focusing on the systems approach to caffeine and health research, where we implement this new omic technology. And so just to begin, as we know, as if you don't know, caffeine is the most widely consumed stimulant in the world, with about 80% of at least Americans consuming some type of caffeine. And there are many different types of sources of caffeine in the diet. Coffee and tea are probably the major contributors of caffeine in the diet, and these are natural sources of caffeine, meaning that they naturally have caffeine and they haven't been added. So when you buy coffee off the shelf, the ingredient list will not include caffeine. It will say just coffee. And there are obviously other sources of caffeine in the diet, and these can include cola, energy, and also chocolate as well. And these often, with the exception of chocolate, these have caffeine added to them, and they're often an ingredient on the list. Mind you that while these are sources of caffeine, you can see that they're quite different. So if you look at coffee, which is naturally basically calorie-free, versus cola, which has a number of calories which correlate with its caffeine content. So these are important things to consider. While this is slightly outdated statistics, it still maintain those numbers today. And basically in the US, coffee is the major contributor of caffeine in the diet for the total population. But as you can see, it varies by age groups, where with the younger groups, sodas and possibly some energy drinks and even milk are major contributors of caffeine. Milk obviously doesn't contain caffeine, but here milk also includes chocolate milk, which has caffeine through its cocoa component. And as we can get older, we see that coffee and tea to some degree become major contributors of caffeine in the diet. So it's no surprise that there's great interest in understanding the role that caffeine or dietary caffeine might play in health. And so that has led to a number of studies looking at caffeine and health in various outcomes. And here's just a brief hierarchy of nutrition research that many of you in this department at Tufts would probably understand or appreciate, where we have more of the lower strength in terms of the relevance and strength of evidence in terms of relevance to humans, but also very important, ranging from non-human studies to meta-analysis and systematic reviews. The meta-analysis systematic reviews often take into consideration all other components of research, including the experimental studies, which we know does have an important place because it contributes to mechanisms. For caffeine and diet, and also for a number of other dietary components, nutritional epi has been a major contributor to understanding the role that caffeine plays in human health, obviously. But it's important to note that these are just associations and do not provide mechanisms where non-human studies might contribute. Given that coffee is a major source of caffeine, it's actually been subject to much research in terms of health, a number of not only hard outcomes such as heart disease and cancers, but also biomarkers of disease, such as glucose and cholesterol. And this was kind of an older, very kind of systematic review, which considered all the data from meta-analysis of coffee and health. And what we see here are some major themes that came out of this systematic review. They found, in general, at least in human studies, they found that coffee of about 3 to 5 cups per day were generally beneficial for a number of outcomes, including mortality, CVD mortality, and incidence cardiovascular disease. Cancer, there is often no relationship, but with lung cancer, there's often an increased risk, and this might be due confounding. I'll discuss some of the limitations of this work later on. Most convincing work comes out of the studies looking at coffee and liver and gastrointestinal outcomes, metabolic disease, and even neurological outcomes. Diabetes and Parkinson's disease have consistently been shown to be while coffee has been shown to be beneficial for diabetes and Parkinson's disease, and that's kind of the really well, not established, but replicated work suggesting that there's a dose response risk reduction for those outcomes. In terms of pregnancy outcomes and even health among children, the data is not very robust, given that it's very difficult to study these populations, and the role that caffeine might play in, say, the development of the brain for young individuals is really difficult to study. And so there are still some open questions in terms of the role that coffee plays in health. And again, these are just based on coffee. But nevertheless, in 2015, coffee, not caffeine specifically, but coffee was added to the US dietary guidelines for the first time. Prior to that, there was no discussion of coffee in the guidelines. And in 2015, based on the literature, predominantly based on the epidemiological studies of coffee, as well as a few randomized control trials looking at biomarkers, they generally concluded that up to 5 cups of coffee per day were safe. Obviously, this is, they even mentioned that it could be beneficial, but this is not, again, these were not recommendations that everyone should be consuming 5 cups of coffee per day. But generally, if you're already consuming that much, you were generally okay, and it would have been fine. But note that 5 cups of coffee per day contains a lot of caffeine. And obviously for some individuals who might be, say, sensitive to caffeine, perhaps 5 cups per day might be too much. And so they're still open for more research and understanding whether these guidelines are in fact relevant to everyone. And with that, I'd like to highlight some of the challenges of epidemiological studies of caffeine and health, partly because much of our guidelines are based on some of these epidemiological studies. These are obviously include classical limitations that are not specific to nutrition or coffee, but include confounding and even measurement error, as we know that our definition of, say, 1 cup of coffee per day can actually quite vary. And you walk into, say, a Starbucks and ask for your standard 8-ounce cup of coffee, it's generally considered a small. And these are kind of things that are not captured very well in some of our dietary assessment tools, at least for not only, coffee is generally captured quite well, but for other sources of caffeine, our ability to measure the amount of caffeine is somewhat limited. And because these are epidemiological studies, it provides no insight to causality or mechanisms. And another important component is individual variation. I'll be discussing much of that further in my talk. And finally, dietary sources of caffeine can vary. Obviously, coffee can come as black or also with added sugar and milk as well. And as many of you might appreciate, you walk into a Starbucks and you can make a pretty fancy cup of coffee nowadays. It's beyond just black, and obviously for individuals who are new to drinking coffee, especially our younger individuals, they generally add things to it to offset the bitterness of coffee. And so those are some of the limitations of the traditional approach to caffeine and health. And so just a little bit about the caffeine absorption and distribution. So some of the pharmacology around caffeine, we actually know quite a lot about how caffeine is metabolized and how we respond to it, partly because it's it's actually a drug, and it's relative because it's so widely available. It's generally safe to study. So generally it's completely absorbed from the stomach and small intestine and into the blood about 45 minutes after intake. Blood levels can peak after, say, 15 minutes to 2 hours. They can vary a little bit based on perhaps the nature of the consumption. That's still not exactly sure about whether consuming it on empty stomach or with food impacts the absorption. It's pretty inconsistent. And following absorption, it's distributed throughout the body and the brain. So it does cross the blood-brain barrier. The metabolism of caffeine is the half-life of caffeine metabolism varies quite considerably. It can range from 2 to 12 hours, and this is kind of a very conservative range. It can actually be a wider range for some outliers. And the metabolism of caffeine is actually well known. And here I just show you the kind of the breakdown of caffeine into its major metabolites. Paraxanthine is the major metabolite. About 80% of caffeine is metabolized into paraxanthine. And while I've indicated a number of enzymes contributing to caffeine metabolism, we do know that over 90%, even 95% of caffeine is metabolized by cytochrome P451A2 or CYP1A2. And much of the variability in caffeine metabolism is probably due to the variability in this particular enzyme, given that it contributes to about 95% of caffeine metabolism. And some environmental factors contribute to the variability in this enzyme. Smoking and heavy coffee intake increase the activity of this particular enzyme. So we do know that much of our epidemiological studies have kind of support this, that there's a strong correlation between smoking and caffeine intake. So smokers tend to consume more caffeine or the dietary source of caffeine. And this is not only possibly a behavioral correlation, but also a biological correlation, because smokers can metabolize caffeine more quickly. They generally will consume more, partly in order to maintain that stimulating effect of caffeine that much of us expect from our dietary source of caffeine. Other factors such as pregnancy, oral contraception, estrogens, obviously kind of related to these three factors, and also liver disease, reduce the activity of CYP1A2, and so these contribute to a lower metabolism of caffeine. So, as you can imagine, women generally have a slower caffeine metabolism, and that kind of changes over the course of their lifespan. And in addition to these environmental factors, genetics also play an important role. Many of us are well aware of the short-term effects that caffeine has on the body. Predominantly, the effect we observe after, say, caffeine, we have that alerting effect or stimulating effect. So it is the most widely consumed psychoactive stimulant in the world. But in addition to these kind of more predominant effects that many of us can experience, there are also other impacts on the body, including the respiratory system and also the heart and other and also the kidney as well. I've also highlighted one component that's really specific to coffee, and it's the increase in intestinal motility and also the microbiome diversity. And I'll be discussing this much later in my talk, just very, very briefly. But for some individuals, they do experience a bowel movement shortly after coffee. This isn't consistent across everyone, but people generally know if they're one of those responders to coffee when they consume it. And that's just something quite unique to coffee that not everyone experiences. These psychostimulant effects and also other physiological responses to caffeine are largely a result of the ability of caffeine to block the adenosine receptors, which in turn block the actions of adenosine and contribute to a number of physiological effects, contributing to, again, for example, our alertness, as well as other, say, respiratory effects. And these are predominantly mediated by the adenosine 1 receptor and the adenosine 2A receptors. To our knowledge, there aren't very many environmental factors that alter the response to caffeine, and so much of this is likely due to genetics. So with that, some of these limitations in epidemiological studies, you can imagine that this individual variation is not well captured in these epidemiological studies, and the dietary sources are obviously a limitation as well. While the common, at least in terms of coffee, to parse between regular coffee and decaf, we generally do say stratified analysis, but still decaffeinated coffee is still quite different from coffee in addition to just caffeine. Actually, there have been data showing that the decaffeination process changes other aspects of coffee as well. So a basic stratified analysis might not do justice to the role that caffeine plays in mediating the role that coffee plays in health. And there are also other factors. And I've had a special interest in individual variation. So given the advancements in this omic technology, including, say, proteomics, metabolomics, and genomics, and basically any type of omics, which are collective technologies used to explore the roles, relationships, and actions of various types of molecules that make up the cells of an organ. These are actually, with advancements over the past 10, 20 years, these have been really more efficient and even inexpensive approaches to really advance the way we look at diet and health, and provides a very high throughput global view, and also hypothesis generating, but also a more targeted hypothesis testing. So with these new technologies, we have opportunities of taking our traditional approach to caffeine research and platforms, because if we capture a particular gene or proteome, this might provide insights into mechanisms. And these omics will ultimately account for that individual variation, for example, genetics. And with accounting for these individual variation provides an opportunity to inform nutritional guidelines and novel preventive strategies that match particular subgroups of the populations who might respond differently based on their exposure. And this in turn will contribute to personalized nutrition. And since maybe around 2006, I've had the opportunity of trying to apply this concept to the study of caffeine, coffee, and health and disease. And I think over the course of my time in the field, we've made some great progress. Not only myself, but others that have applied these types of omic-technologies to coffee and health research. And the next few slides, I'll be discussing my work on the genetics, proteome, and metabolomics. Others have contributed to the field looking at the epigenetics, transcriptomics, and the microbiome, but I'll be discussing some of the work that I've personally done in this field. So as mentioned, since the sequencing of the human genome, the advancements in genetic technology has really given us the ability to kind of understand the role that genetics play in our dietary behaviors and even our response to diet. And this has really ultimately given rise to the genome-wide association studies, which are now kind of a major tool in our epidemiological toolbox. Since maybe the last decade, they've been pretty consistent in the research, in the literature. So when I began my postdoc at Harvard School of Health, just briefly, when I'm talking about genetics going forward, I'm really talking about the common single nucleotide polymorphisms. And these occur in roughly over 1% of the population. And so I won't be discussing mutations, but really polymorphisms that can be that are found in most populations. And so the genome-wide association study approach, I've had the opportunity of applying this to coffee and caffeine drinking behavior, as well as circulating caffeine metabolites. I was fortunate enough to begin my postdoc right about the time that GWAS were being introduced. So I had the opportunity of gaining some great practical experience in this field, and have had the opportunity of applying this technology since around 2010 to these particular outcomes. And I've worked with a number of large consortium and other population studies, some including, say, the Nurses Health Study and also the UK Biobank and others around the world. And myself, as well as others, have contributed to the major genetic factors underlying not only our drinking behavior, so So what factors contribute to our propensity to consume caffeine, but also circulating caffeine metabolites, which provide insight to caffeine metabolism. And we've identified a nice handful of genetic variants. And while I won't go into the details of each of these studies, I will summarize our basic findings for these genome-wide association studies. And here I just provide you the variants, not specific variants or DB SNPs, which is how we typically tag these SNPs, but rather the closest gene in which these variants reside. And here I just provide you kind of a snapshot of, for example, we have a variant in the AHR receptor, which the one particular variant leads to, is associated with higher circulating levels of caffeine in the blood, while the same variant, it contributes to a lower caffeine consumption behavior. And in addition to aryl hydrocarbon receptor, we also see CYP1A2 among these. And these are actually ranked in their order of effect sizes. And so CYP1A2 was kind of reassuring to see on our list because it is a known enzyme involved in caffeine metabolism. It's actually one of the enzymes I studied even prior to genome-wide association studies. And so it kind of tells us that these GWAS are quite robust and are actually capturing some very useful and biologically plausible variants. AHR wasn't originally thought to be involved in the main pathway of caffeine metabolism, but is a major gene involved in the regulation of CYP1A2 expression. Other variants are also implicated in caffeine metabolism. So caffeine metabolism pharmacokinetics include probably our major variants that have the largest effect sizes. And as you can see, this consistently show that variants that are correlated with higher circulating caffeine metabolism are correlated with reduction in caffeine consumption behavior. So I'll discuss that briefly later on, but we also find ones that are related to the response to caffeine, the adenosine 2A receptors, and also BDNF and SLC6A4. But I'd like to focus on pharmacokinetics because they're the major, they're the predominant and most robustly replicated SNPs related to caffeine traits. And the interesting pattern that we see here with circulating caffeine metabolites and the actual behavior is in the opposite direction. So it's kind of interesting. It's actually demonstrates some type of titrating effect. So I'll discuss those a little bit later on, but there are also other variants that don't necessarily fall in pharmacokinetics and dynamics umbrellas, but they're actually pleiotropic in that they're related to a number of other traits. So GCKR and MLX-IPL, for example, are linked to glucose and also lipid metabolism. So how they relate to caffeine metabolism or behavior is really unknown. And so much of my work going forward has really been focusing on ones that have a biological plausibility. And getting back to what I was mentioning about the direction of effects of these would suggest that we're showing here that individuals are naturally kind of titrating their caffeine consumption behaviors in order to match their genetic variants. So, for example, individuals who have genetic variants related to low caffeine metabolism, as suggested by higher circulating caffeine in their blood, basically when they consume a cup of coffee or tea, caffeine circulating in their blood is maintained a little higher, and so they naturally reduce their dietary caffeine intake. Whereas the opposite would be observed for someone with variants related to increased caffeine metabolism. They will naturally consume more caffeine. So what we're kind of seeing at a population level is this natural titrating effect of caffeine. That kind of just reiterates I guess how many people are well aware of the effects of caffeine in terms of the psychostimulant component. And so this just it's kind of scary at the same time, suggesting that people are well aware of the effects of caffeine and modify their behaviors in that order. And this titrating effect is actually not new. It was actually demonstrated much earlier, even in clinical trials. This is just one example of a couple by the same group where they took a kind of a working or office environment and altered the caffeine concentration of the coffee pot in the kitchen. And what they found is that when they lowered the concentration of caffeine in the coffee, they found that people were consuming much more coffee. So they obviously weren't feeling the alertness that they typically experience with their general 1 cup of coffee per day. And so they were kind of altering their behaviors accordingly. So it's kind of interesting that people are really well aware of their responses to caffeine. And here's just another demonstration of how this works, looking at just taking a CYP1A2, for example. This is really just a cartoon demonstrating how this would work. So holding 1 cup of coffee, for example, steady, we find that individuals genotype that correspond to rapid caffeine metabolism, over time, they'll metabolize caffeine quite quickly. And so their overall exposure, their physiological exposure to caffeine is much less compared to someone with a genotype corresponding to slow caffeine metabolism. You can see that the caffeine levels here, concentration, is maintained a little higher throughout over time. And in order to maintain this happy medium in terms of psychostimulant effects of caffeine, you can imagine that individuals who are metabolizing caffeine quite quickly only experience that kind of optimal amount in a kind of a smaller time frame. However, because humans are so complex, what we ultimately see is that we actually alter our actual drinking behavior, as mentioned in my previous slide, that these individuals are actually consuming more coffee or other dietary source of caffeine to kind of accommodate that rapid caffeine metabolism. And individuals with slow caffeine metabolism naturally are consuming much less dietary caffeine. But again, because humans are so complex, we're not just seeing alterations in caffeine behavior, but also seeing alterations in other things. So for example, we find that individuals, on average, individuals with a genotype corresponding to an increased caffeine metabolism, not only are they consuming more coffee, they're also preferring their coffee black. And they're also more likely to be consumers of decaf. So it's possible that they've been naturally cutting back on their coffee because they think they're consuming so much, but they've kind of acquired a taste for that bitterness of coffee. They also enjoy much more dark chocolate compared to someone with slower caffeine metabolism. We find that those with slower caffeine metabolism actually prefer kind of lower concentration sources of dietary caffeine. So for example, our sodas, they also consume more water as well. And they also like to add sweeteners to their coffee. So besides being different in terms of caffeine metabolism, we also see other behaviors that are altered as well. And this is important to take into consideration when I discuss some future applications of this work. And so how can we actually use these genetic findings? We can use it in terms of risk assessment or intervention design. So we can maybe modify our randomized control trials based on genotype, also provide biological and mechanistic insight. And this is really where much of my research has been focused on. And you can also imagine the food and drug and manufacturer and marketing might also have some interest in knowing these genetic differences. And given that, as I mentioned, that it looks like these genetic variants might alter our behavior, they might also kind of contribute to what some people argue is that caffeine is potential drug of dependence. We talk about this really anecdotally about how people being addicted to caffeine, but interestingly, the US and even kind of international classifications of diseases do not really consider caffeine a drug of dependence. Sorry, a US does not consider caffeine a drug of dependence. However, other guidelines or other classification systems do, and so it kind of varies. But although caffeine isn't considered a drug of dependence, they do recognize caffeine withdrawal symptoms because we know that these withdrawal symptoms are often a symptom of an addiction. And so these are actually acknowledged, but caffeine is, however, not considered a drug of dependence. This might partly be due because flagging caffeine as a drug of dependence might say trivialize real addictions such as tobacco or even cocaine. But some might argue against this, and there are kind of a group of individuals who really do believe that caffeine should be added to this list of drug of dependence. But you can imagine that with this genetic research, it suggests that perhaps some people might actually be more prone to caffeine dependence. I've had a special interest in looking at gene-diet interactions, so specifically SNP and coffee and caffeine interactions. And this actually began during as a graduate student at University of Toronto, where I looked at the interaction between variants CYP1A2 and coffee consumption and risk of myocardial infarction. And this was published really a long time ago, in 2006. And it's often a paper that's quite often cited for some of these direct to consumer genotyping services. Many of you are likely aware of 23andMe. And for a long time, they highlighted some of these genetic variants related to caffeine. And they often cited my work as a PhD student looking at the modification, where we actually showed that individuals with the CYP1A2 genotype corresponding to low caffeine metabolism were actually at an increased risk of myocardial infarction when they consumed high amounts of coffee. Now, I'd just like to out myself. Since this time, there's really not been very strong replication of this work, and this was actually based on the Costa Rican population. And so, obviously, I think now with much more work in the area of gene-diet interactions, there's been a greater push for a replication. And so that's at the time, gene-diet interactions in 2006, they were kind of these candidate studies were published quite often. I think nowadays more people are becoming more skeptical about the ability to replicate some of this work. And so that's just kind of I think in terms of advancements in the research of genetic epidemiology, I think the replication becomes much more important. In addition to that earlier work, I've also applied these gene-diet interactions and the concepts of cardiometabolic biomarkers, mortality, and even COVID-19. And more recently, I've explored the area of aging, looking at predominantly cognitive ability as well as dementia. And this is more of my more recent funded research. So I've kind of moved away from cardiometabolic outcomes and kind of focused in on cognitive function, partly because caffeine has been implicated in cognitive ability as well as dementia. Besides gene-diet interactions, Mendelian randomizations that have utilized these caffeine variants as instrumental variables is quite common. Although I'm not a fan of these, and I'll discuss why. But basically, as for some of you who might not be aware, but in the Mendelian randomization, it kind of acts as almost like a randomized control trial in that it uses the genetic variant as a marker of exposure. And obviously based on a number of assumptions that these genetic variants are not related to, say, confounders or directly related to outcomes, they can serve as a proxy for our exposures. And therefore, if a direct relationship is found between variants and outcomes, would be an indicator of a causal relationship. Because as you know, our standard epidemiological studies cannot presume causality. So just to demonstrate this again, as we know with randomized control trials, individuals are randomly assigned to a particular arm, in this case, say, for example, caffeine to control, and they are then followed up, assuming that confounders are kept equal between groups. And disease outcomes are measured, and then statistical analyses are conducted to assess the role that caffeine played in contributing to these outcomes. Mendelian randomizations take a slightly different approach, and they take the natural stratification allowed by genetics. And so we have the population stratified by a caffeine genetic variant, such as CYP1A2, and then we follow them over time, particular disease outcomes, and also look at statistical tests. This would be very useful for example, diseases that have a longer incubation time, say for cancers. Obviously, a randomized control trial waiting for cancer cases can become very inefficient. So Mendelian randomizations have been thought to serve as a very efficient design to understand the causality. But again, there are certain assumptions, one specifically being these confounders being equal, that allow these Mendelian randomizations to be really robust methods. But as I already mentioned, that while these variants can alter caffeine exposure, we do know that other dietary factors are changing as well. The type of coffee they're consuming and even how they prepare their coffee. So, for example, if these individuals on average are metabolizing caffeine quickly or consuming more coffee, they're also consuming their coffee black. And so we really don't know if it's coffee per se or other or caffeine or other components in these dietary sources. And so these are kind of things that we need to be aware of when we critically evaluate these Mendelian randomization studies. And so I've been a co-author on some of this work and have highlighted in the discussion the limitations methods that these are obviously there's some limitations in these Mendelian randomizations. And so nevertheless, we see a lot of these in literature now. Oops. And so moving away from the genome, I'd like to focus briefly on the work I've done in metabolomics. And up until around 2016, there have been a number of cross-sectional studies. And these have identified biologically plausible correlations with coffee consumption. These have been predominantly population-based studies, and they found that circulating levels of methylxanthines, for example, are caffeine metabolites, and also trigonelline and other compounds that are specific to coffee. These are actually correlated with coffee consumption behaviors. So these are kind of known metabolites. And so that just kind of tells you that this metabolomics of coffee consumption in a population setting is quite useful and is actually yielding some plausible findings. Obviously, again, these are cross-sectional, and so obviously have their limitations. So I had the interest in applying this metabolomic approach to a coffee trial of coffee. And so I reached out to a Finnish group who had already performed a coffee trial, and it was published already in 2010. And this was a 3-stage clinical trial, so it wasn't randomized. And so this is partly because it's very difficult to dramatically alter individuals' coffee consumption behavior. So telling them to consume and then abstain from coffee is very difficult. Some of my colleagues that have in this area are kind of are very well aware of these issues with coffee trials. But nevertheless, I took advantage of this particular trial where they asked individuals first to abstain from coffee for a full month, then consume 4 cups of coffee per day for another month, and then for another month, 8 cups of coffee per day. And 8 cups of coffee per day sounds like a lot. Obviously, it's more than our US guidelines suggesting. But take into account that this is actually a Finnish group. Finland is actually among the highest coffee consumers in the world. And so 8 cups per day isn't too abnormal, but definitely different from the average American. And all these individuals are at high risk of type 2 diabetes. And the ultimate outcome, the primary outcomes of this trial were really the glucose, measures of glucose homeostasis. But nevertheless, it gave us the opportunity to acquire blood samples and run metabolite profiling. So I reached out to these investigators, and I used their available plasma. And using the Metabolon platform, which captured around 733 metabolites from various pathways, we conducted a metabolomic study of basically coffee response in this trial. And we found that over 115 metabolites were altered with coffee consumption. And while I won't go into the details of all 115, I show you here the major pathways where these metabolites were found. And the ones that are in these little stars here are pathways in which we found a significant enrichment. First, focusing in on the xanthine metabolism, there were basically all 14 metabolites of this pathway increased as coffee consumption increased over time. And these include our caffeine metabolites. So this was a good positive control. All those caffeine metabolites increased in the blood. This is also was also of interest, the benzoate metabolism. And as I mentioned, the coffee has an impact on the microbiome, and many of the say polyphenols that are found in coffee are metabolized by the gut microbiome. And what we found is that these metabolites were also enriched, also increased. And so this was kind of a positive control, as well, as we were actually expecting these to increase in addition. But we found additional pathways that were never really implicated in coffee consumption or response. And I'll focus just on one, and that's the endocannabinoid pathway. We found 4 of the approximately, I think, 6 metabolites we found were decreased with coffee consumption. Now, these endocannabinoids are natural cannabinoids that are produced by the body. And it was interesting to find that they actually went down. And I reached out to some experts in the field, and they indicated that usually the level of these endocannabinoids decrease in response to stress. So perhaps over time, with increasing coffee consumption, our stress levels were maybe increasing and altered the activity or the levels of these metabolites. But that's obviously something new that we've never seen with coffee. And the system is especially interesting given that it's been implicated in so many different pathways and even health outcomes. In terms of obesity or even our appetite, we do know that our natural cannabinoids, such as cannabis, lead to an increase in food intake, and that's with the increased cannabinoids in the system. And so it'll be interesting that we find that coffee actually lowers endocannabinoid levels. And so there has been some data suggesting that coffee alters or inhibits appetite, and perhaps this could be one mechanism by which this happens. But nevertheless, it's very interesting that we find that this particular pathway is altered, and it just demonstrates that not only the metabolomic approach identifies biologically plausible pathways and metabolites linked to our response to diet, but also potentially some new pathways. So for, say, hypothesis generating and further follow-up in studies. I haven't really followed up on these particular metabolites yet, but I look forward to doing that in the future. Taking the same 3-stage clinical trial, I also conducted a lipidomic study, which is also kind of related to metabolites, and also using the Metabolon platform, and looked at over 800 lipid species from different families, 14 different lipid classes. And while we did not find a large number of significant lipids, like as we did with metabolites, we did find one particular class of enrichment. We found that coffee generally led to a reduction in some key phosphatility choline. These are the role that these, or the impact that lowering the levels of these lipids have on health are really unclear. In the area of cardiovascular disease and cardiometabolic outcomes, generally a reduction in these is a good sign, because these are often a product of, say, LDL oxidation. And so what we see with increased coffee consumption, perhaps LDL oxidation is reduced, and so we see a lower concentration of these lipids. However, in, say, for example, infectious disease, these a reduction in LPCs is often not a good thing. But however, it's still unclear the role that these have in health. But nevertheless, this is a kind of a novel finding linking coffee and the alteration of the circulating levels of these lipids. And very briefly, I also performed more of a targeted proteomics study, again, using the same 3-stage clinical trial, using the Olink platform, which is really targeted. Unlike the metabolomics field, the proteomics field took a little longer in terms of coming up with high throughput measures of the proteome. And so at this time, we used more of a targeted approach. We looked at proteins capturing cardiovascular, immunity, and also neurological systems. I had a special interest in these, obviously based on my prior research on coffee and these outcomes. And from there, we only found 2 significant changes after we counted for, obviously, multiple testing and the number of proteins. We found that that as coffee increased, the levels of carboxypeptidase M and neutral ceramidase also increased. These have unique roles in protein modification and also lipid metabolism, but their impact, their direct kind of relationship with coffee consumption response is unclear. And so again, these are novel findings that are worthy of follow-up. And one slide on the microbiome. I've not done work on this, but there's been great interest in this. And as already mentioned in my metabolomic studies and even the cross-sectional studies showing that coffee appears to have more, has a direct interaction with the gut microbiome, partly through its polyphenol content. And there have actually been population studies that have really honed in on a number of dietary factors, and they show that among the dietary factors, coffee, I'm also mentioning soda with sugar, has a marked impact on the gut microbiome. It contributes to, is a major contributor to the variation in the gut microbiome. And as you can see, looking at coffee, a major source of caffeine, and soda with sugar, also another source of caffeine, they have actually opposite impact on the gut. Yeah, that's sure that kind of threw me off. Okay, here we go again. Just briefly summarizing my work again. These are kind of the steps that have been taken in order to apply this new omic technology to really advance the way we look at coffee and even caffeine and health. And I think applying these technologies to not only these dietary beverages, but also diet in general, we're really going to be able to really advance our dietary guidelines or recommendations for individuals, taking into account some individual variation, which will ultimately lead to optimal or guidelines for optimal nutrition. And with that, I'd just like to conclude by acknowledging mentors and collaborators, large collaborators from my consortium work looking at the genetics of coffee and caffeine traits, but also the mentors, my PhD supervisor back in the day, Frank Hu and Peter Kraft from Harvard School of Public Health. As I mentioned, most of you probably are aware of them. They were kind of major players in my experience in the area of omics. And then my close collaborators, Rob Van Dam and the late Martha Claire Morris, who unfortunately passed. And with that, I'm open to any questions.
Thank you. What a fascinating talk. We can't, it's hard and virtually to give the proper round of applause. So I think you'll have to take our virtual round of applause. Really, that was just such a variety of interesting topics. So thank you so much. Ed, do you want to kick us off with a question? And everyone, please feel free to raise your hand. And we'll go for the questions and answers now.
Well, if you insist, thank you for a really interesting presentation.
Thanks.
So I was thinking about the label of the coffee that's sitting in my kitchen. It has tasting notes of things. It says things like, notes or hints of lychee, pine nuts, and motor oil, things like that. So I've never tasted those things, but I'm assured. Actually, one of our alumni is an executive at Keurig, and she assures me that some people can actually taste those things in coffee. So the question is, does people's ability to taste those more subtle flavors have anything to do with their consumption, either the amount or how they drink their coffee, and are there associated metabolic phenomena?
Yeah, these are great questions. And actually, when I started pursuing GWAS and coffee consumption, I thought our top hits were going to be related to taste. Because some people are prone to the bitterness of coffee, but at the same time, if we're using these population-based studies, if people don't like the taste of coffee but want the caffeine, they're going to be altering it with, say, milk and sugar. And But as going back to your taste, I thought taste factors would come up because we do know that there are well-known polymorphisms in the taste receptors. That's almost like a classic genetic variance in our ability to different tastes. And so we actually did some studies looking at that particular taste receptor, and we did find that it does impact the type of coffee you consume. So, but yeah, getting to your point, there's definitely everyone's experience of foods is quite different based on the different strengths and responsiveness of their taste receptors. It's pretty remarkable that, say, you know, going back to those taste receptors with bitterness, for example, people, a child, their experience with broccoli can be quite different from another child's. And if they say it tastes like crap, other ones are like, I don't taste it at all. Like, you know, I think people now kind of appreciate that people do have differences in taste, and it's really genetics that underlie these factors. But in terms of coffee and caffeine consumption behavior, the most strongest variants were related to caffeine metabolism. So at least with coffee and caffeine, major dietary source of caffeine, it's those genetic factors that are really, people are just consuming these beverages based on that caffeine.
That is just fascinating. And can I ask, other people, please feel free to put your hands up. Can I ask, I'm curious about, in terms of advice to the population, I mean, it sounds like people are pretty self-aware, and maybe people don't need, you know, their own genome sequenced to already receive some advice. I'm just curious how you would propose to do something like this, to say to people, okay, if you tend to have these, if you can relate to X, then you should do Y. If you can relate to something, I don't know. I'm curious about that, and how you think that would go forward in terms of dietary.
Yeah, okay. Right. So this is obviously, in my opinion, I think with a lot of the research in coffee, at least with coffee, black coffee specifically, I think there seems to be much more benefit to be gained. And with my genetic research, it suggests that people are naturally altering their behaviors in order to align with their genetics, and so that they can tolerate caffeine. So if you see someone drinking a lot of coffee and you're like, my goodness, that's crazy. I can only drink 1 cup of coffee per day. You're different. You're genetically different. And obviously over time, and I've discussed this with other people do acquire a tolerance, and they become, you know, they can consume more and more. And so if they're as a peace of mind, they want to cut back, then sure, go ahead and cut back. But I think if you're already consuming, say, 5 cups of coffee per day, which sounds like a lot, but your health is fine, then I wouldn't go crazy and try to cut back. I've been asked to do a number of like podcasts and interviews with people, and they one of the introductions to kind of inviting me, they're saying there, I want to start, you know, I had this group that we kind of want to know ways in which we can cut back on our caffeine. And so my first question was, why are you trying to cut back on caffeine? I think if you're healthy and you're already consuming that much, I think it's fine. And the thought of cutting back on caffeine is actually quite hard for a lot of people. And so I think there are obviously other things in your diet that you can modify that would probably take more priority, maybe cutting back on your sugars. I think those are those are the hangy fruit that you should really addressing. I wouldn't lose sleep over, no pun intended, I wouldn't lose sleep over the amount of coffee you're consuming.
That's excellent.
That's my opinion.
Yeah. B, do you you can unmute and ask your question. Go ahead.
Oh, well, no, it seemed from what you were presenting that I mean, you got a lot into the individualized metabolism, but overall, I didn't see a whole lot of, oh, geez, this is a big public health problem. We've really got to stop people from. I heard some benefits, longevity, and so on. And I didn't hear all that much about risks. So I'm just wondering why there would be a focus on figuring out how to get people to cut back.
Yeah, I think it's, yeah, obviously, I guess my research really kind of does bias towards the benefits, and as already suggested by my kind of advice, but I think most of the work that we've been doing is really based on adults. There are certain populations like younger children and infants who we really don't know the impact that caffeine has on, say, the developing brain. So that's why there's great interest in that. And obviously, it's very difficult to study, say, pregnant women and children. And there's concern partly because the industry has come up with very novel ways of increasing caffeine consumption, and they're targeting younger individuals. And these younger, like 9, 10-year-olds, they're not going to be drinking coffee. They're drinking other things like soda. They've got caffeine in it, but we also know soda has other things. And if these kids are kind of start to experience the effects of caffeine have and become really kind of not addicted, but tend to favor those kind of beverages, they're not only consuming caffeine, they're consuming a lot of soda. They might never, over time, maybe they'll never become coffee drinkers. So they're missing the benefits of, say, other natural sources of caffeine. I think the concern is really getting these kids hooked on to caffeine per se and not other natural sources of caffeine.
I had a question, I'm sorry. I wanted to know, is there a difference between like espresso and coffee, or was your research just focused on coffee? Because there's a lot of myths about espresso having less caffeine than actual regular coffee. So
when it comes to the form of coffee, and at least in terms of health, the major difference is the impact that coffee has on your cholesterol levels. And it's really not caffeine per se. So for example, this was actually shown in randomized control trials. So much of the American coffee is generally filtered. Our ground coffee, espresso, I think, is unique in that it's not filtered. I believe espresso is one of those. The boiled coffee, so your Turkish coffee, your French pressed, these have not been filtered. And what's interesting with the filtered coffee, it extracts the lipid components or the diterpenes found in coffee. And these diterpenes have been shown to increase cholesterol levels. So individuals who are consuming these very rich sources of really rich types of coffee, such as your espressos, your French press, which are actually becoming more popular, these are actually not so good for your cholesterol levels, especially if you're consuming them in large quantities. So if there's any way you can filter that coffee, that would be great, because that's been shown. These diterpenes, these kind of fats that we find in coffee that are naturally filtered in much of our coffees, they persist in those kind of fancier coffees. So that's something to consider. The other independent of caffeine, I think they really kind of work the same way. In much of the FFQs, some of the say espresso, I think, is more common, say, for example, in the Mediterranean. And so those FFQs do really distinguish between those. And so they actually account for that when they derive caffeine. And so that's taken into account some of these population studies that I've conducted. But yeah, that's just kind of in terms of the different impact that coffee has on health. The form, it really has an impact on those cholesterol levels.
of course, I love espresso. So
Me
thank you.
too.
Great.
Well, maybe one a day is fine, but the US guidelines of 5 cups of coffee per day are not talking about 5 espressos a day, because that's very different.
Wow.
Thank you.
Well, thank you. I think we probably have to let people go now because we've hit time, but this is really fascinating. So thank you
Yeah,
so
if
much.
anyone has any other questions, you feel free to email me. Yeah, I'm on Northwestern, pretty easy to find me. You can just Google Marilyn Cornelis nutrition caffeine, and yeah, I come up someplace.
Fantastic. Thank you for the offer. I do hope people reach out, and it looks like we'll actually be collaborating on a few different projects anyway with Tufts. So looking forward to being in touch, and thank you so much for joining us.
Thanks for the invitation. Much appreciated. Have a good day. Take care.